Hello, everybody, and welcome to the live Q&A session. So thank you for joining us today, and thank you, Emma, for joining us and for what was a very, very interesting seminar. So thank you so much for being here today and taking the time to field questions. Just before we kick off with questions, I'd just like to remind everyone that you can submit questions through the online portal. So please do that. It's a great opportunity to quiz Emma. And also, if you'd like to vote on questions, you can do so, and that prioritizes the way that we ask them as well. So without further ado, Emma, I'll just go right to the top ranked question. And that is, have you studied differences in baseline laboratory indices, such as interleukin profiles, among the two SRS groups?
Yeah, so that's something that our collaborator, Julian Knight, and his team have been working on recently, and they've generated some cytokine data for the full cohort. Historically, it's been quite challenging to use a single biomarker, such as a single cytokine, to sort of differentiate sepsis in terms of survival and things like that. And again, we see that we're not able to differentiate these particular SRS groups very well with the cytokine data alone. But actually, there are two slightly independent groups that come out from the cytokine data. So there's a manuscript under review at the moment. So watch this space for a bit more information on that side.
And is that based on a single cytokine, or is it kind of a mixture of multiple different cytokines that give you the ability to discriminate different groups?
That's based on a multiple, so it's sort of a panel of a small number, but a panel of cytokines for that.
Okay, great, thank you.
Thank you, Emma. Another question is if you know if the two SRS groups have a similar count for the different white blood cell types?
Yeah, so we do see that there are slightly different cell counts between the two groups, and that was one of our initial questions, was whether that was driving all of the differential expression that we're observing. When we include these cell counts in our model, though, we still see a very high correlation in terms of the genes that are differentially expressed after accounting for cell count. So we think that it's likely that they will have an impact, but that the majority of the signal is still coming from other sources as well.
Thank you.
Thanks, Emma. We've got a question that says, are the genes differentially expressed within the SRS groups known to play a role in other immune-related conditions, for example, autoimmune disease?
Yeah, so this is an area that we haven't spent enough time yet exploring, but it's certainly something that we're really interested in thinking about, what the different sort of components of these signatures and what might make them up. Although not sort of directly sort of related to that question, one area we have been beginning to think about a bit is endotoxin tolerance. So this occurs when you have a repeat exposure to the endotoxin. So for example, lipopolysaccharide, that's a bacterial endotoxin. And so we see that there is enrichment of some genes that are known to be important in the endotoxin tolerance that are differentially expressed between these two SRS groups. So this is definitely an area that we're looking to expand and to think about some other sort of components that might be sort of contributing to this signal, and really trying to sort of disentangle what that might be. And I think certainly it's going to be interesting to have the opportunity to try and integrate other immune-related diseases or autoimmune diseases and things like that as well, to see if there are particular features that overlap.
Thank you.
Great, thanks, Emma. And I'm going to follow up a little bit on Roser's question, because there was another similar one about that. Is I guess, do you know the cell types in your interleukin population? I guess the proportion of those different cell types, and whether any of them really underpin this, I guess, this SRS set of genes. And what are your thoughts about using things like single cell RNA sequencing to really deconvolute this?
Yeah, so we have some information available through the hospital records on quite broad cell populations, so lymphocytes, polynucleocytes and monocytes. We're beginning to sort of try and integrate that into some of our models to understand which are the cell types that are potentially contributing to that. We've been exploring some of the deconvolution methods, so some of the methods that are out there now that can be used to try and predict the proportion of cell types from your gene expression data. One of our concerns with sepsis is that it's obviously the huge amount of immune dysregulation, and so the genes that may be used to impute what your cell proportions are potentially being dysregulated, so it may or may not be the most sort of robust way of really getting an estimate. One exciting thing is that the GAINS study is continuing to recruit, so we now have GAINS 2. So Julian and his team are collecting samples from a smaller number of individuals, but now focusing on being able to do much deeper immunophenotyping on those individuals. So we will be generating things like single cell RNA sequencing for a smaller cohort. And we think that's going to be really exciting to then try and link in with our much larger bulk RNA sequencing data set. And for example, we may be in a better position then to start trying to deconvolute some of these signatures from the bulk sequencing based on a smaller number of single cell RNA sequencing data sets.
Because presumably you know what the cell types that express at least some of the key marker genes that you see in your signatures, but I guess the caveat being the point you made that some of these are actually dysregulated in the disease itself, so that may confound that analysis.
Yeah, and I think, you know, trying to make use of the resources like the Human Cell Atlas are going to be really exciting to sort of see, you know, what's happening generally in a sort of healthy setting or a non-sepsis setting, and then seeing what we observe in the disease setting as well, and sort of working out, you know, can we use healthy cohorts as a reference, or do we need to be generating these data sets in a clinical setting to really get the best understanding for our disease?
Thank you.
Yeah, I will follow up a bit on this as well. Do you think that there are multiple cell types that are involved in this SRS signature? For example, do you see any ligand receptor pairs differentially expressed in the groups that may indicate that, yeah, there is a network of cells really creating this dysregulation?
Yeah, so we haven't specifically looked at the receptor ligand pairs, but it's definitely a really good idea to start focusing on some of that. And you know, I think given that from the sort of quite crude cell data that we have included, the fact that it isn't one particular type that's standing out at the moment as necessarily driving all the signal, then I think it is likely that there is going to be sort of multiple cell types that are potentially contributing. And some of the differentially expressed genes that we observe suggest things like T cell exhaustion could be important, distinction between the two groups. So we know that that might be important. There's other evidence to suggest that potentially neutrophils might be important as well. So again, I think there's potentially going to be quite a few different cell types, and it's going to be a good challenge trying to disentangle sort of some of the signals there to understand where they're coming from.
Thank you.
Got changing tack slightly. There's a question about whether you're planning to use epigenomics tools such as ATAC-seq to understand the impact of non-coding SNPs on your gene expression.
Yeah, so at the moment we're planning the large EQTL mapping, which will enable us to get an idea of the non-coding variants that are potentially having an impact on gene regulation across the genome. And certainly once that's complete, it'll be really interesting to then try and layer on some additional information, particularly sort of trying to find map in a particular locus which of the variants are possibly causative in terms of the regulation of that gene expression. I think it's still quite challenging to generate some of these data sets on the clinical samples because of the numbers of cells that are required and being able to get access to the fresh material. But again, it would be something really interesting to be able to try and integrate potentially some healthy donor cohorts to sort of understand if that could be informative for this kind of interpretation as well.
Good. Hi Emma. So, you know, in your presentation, you're very focused on the population that are actually, I guess, have very severe disease and are admitted into an intensive care unit, which is obviously very important. But actually, I don't know how many go into intensive care, but there's clearly a large population that never, their disease never gets that severe. And I just wondered if there was an opportunity also to kind of look at, I guess, markers for disease severity as opposed to outcome as well, and whether you're thinking about that, or is that practically difficult because you would need to sample very large populations, for example?
This is definitely a question that we're really interested in, and actually we've got quite a new collaboration with the Bioresource in Adult Infectious Disease. So this has been set up to recruit adult patients when they present to the emergency room with a suspected infection. So we are taking a sort of step back. So we're sort of going to that earlier time point, potentially in someone's disease progression. Sepsis is heterogeneous, and even on the intensive care unit is heterogeneous. So this new cohort is going to be even more so. So there will be challenges there, but we think this is a really exciting opportunity. We've at the Sanger Institute, we've recently generated RNA sequencing on around 1,700 individuals that have been recruited to Imperial or UCL in emergency room. And these individuals are either individuals that have had a confirmed respiratory infection, a confirmed urinary infection, or no infection could then be identified. So they can act as a sort of control in terms of their people where an infection is suspected, but we can't then figure out what it is. One of our key questions is whether or not we do observe these sepsis response signatures at this earlier time point, and obviously bearing in mind that there's going to be a real spectrum of individuals in terms of severity. But we will also then have clinical information about whether or not individuals go on to develop sepsis to the severity where they then have to be admitted to the intensive care, or if they're sick enough to be admitted to a ward, or if they're actually able to be discharged home. So there's the second half is then, as you alluded to, is to then see whether there are potentially other signatures that might be helpful for distinguishing those who are on a trajectory where they may be more likely to be developing a more severe outcome, and those that, you know, you may be sort of quite happy to be able to discharge them home and be able to manage them from the community.
And how large is that cohort? Is that the 1,700, or that would actually?
Yeah,
that's the 1,700. Okay.
And the really sort of exciting thing about this cohort is that the BioAid Bioresource is aiming to recruit 10,000 individuals. So while we've generated data for this first sort of subset, there'll be lots of exciting opportunities to be able to make use of additional samples in the future to do any follow-up. So if we find that there are some particular signatures in particular groups of individuals, we can go back to the bioresource and be able to do some validation.
Okay, thank you so much.
So is SRS being used to inform clinical intervention? How stable is the signature over the course of patient treatment, days 1, 3, and 5, following this ICU admission?
Yeah, so it's not being used at the moment in the clinic, but it's something that we're really interested in thinking how we can make this as translational as possible. From some initial work that we've done with the microarray data, we have patients with fecal peritonitis, and that's where there's a perforation in the gut. So it's slightly easier to know the point at which somebody has had the injury that means they're then going on to develop sepsis. With the community-acquired pneumonia patients, it's quite challenging to know at what point in their disease progression they're admitted to the intensive care unit, because there's lots of factors that could affect. So one person's day one might be someone else's day one. So from the fecal peritonitis patients, it was a very small number where we had multiple samples available. We saw that there was a good chunk of individuals that stay in one endotype, so they stay in SRS1 or SRS2, but there are some that do move, and generally if they move, they tend to move from SRS1, which is the poorer outcome, into SRS2. This could, of course, be a sampling bias, because to be able to have a sample, you have to be surviving those first 5 days. So we have to bear that in mind. In our new RNA sequencing data, we have about 190 patients that have multiple time points available. So this is something that we really want to explore a bit more. And it's one of our key questions, is how much do these SRS groups change over time? If they do change, it can still actually be very informative, because it might be then quite a useful monitoring biomarker. So rather than necessarily a biomarker that sort of shifts you into one group or another in terms of treatment, it might be a way of monitoring if treatment is working. And we're working with our collaborators, Tony Gordon and David Ancliffe. They looked at these SRS groups in their VANISH clinical trial, and they've now generated some data with pre and post time points for looking at so we can look at SRS group in response to treatment and see whether or not it's changing. And again, that will be really interesting to know if we can modify somebody's SRS group, then suddenly that's an interesting sort of angle in terms of thinking about other treatments for the treatment of sepsis.
Thank you.
Somewhat related to that, it was whether HIF1 alpha could be a potential biomarker or a therapeutic target in sepsis. Has work been done in that area?
I don't know the literature well enough for that particular area, but I know that it has been a sort of an area of interest in terms of research in sepsis in general. And certainly, again, I think the fact that we're seeing a difference between these two groups in that gene in terms of differential expression, and we also see that there is an EQTL for that gene as well. It's, you know, it does suggest there's something interesting going on there in terms of regulation and its potential sort of downstream impact on many genes. So I think we're still a sort of way away from being able to have necessarily new targets, but this is certainly a good way of starting that process and thinking about how patients can be stratified if you do have an existing drug that you want to test, but also then potentially the opportunity to have some new targets as well.
So there is a question about COVID-19. So sepsis shows some similar features to severe COVID-19, like demographic risk, hypoxia, netosis. Is this a research avenue you are investigating?
Yeah, so it's certainly obviously been a really interesting few months for us. You know, our main research focus is heterogeneity and response to infection, and you know we've seen in the world around us just what an impact that can have, looking at the different individuals who've been hospitalised with COVID. We're working with the collaborators in Oxford who've set up a big study on recruiting COVID patients called COMBAT, and so we've been involved with them of thinking about how to look at the similarities and differences between sepsis and COVID, and obviously it's a really interesting question because you know we do have some individuals in our cohort who have a viral infection, and COVID is you know a unique opportunity to look at the response to a single viral infection and really sort of understand why there can be variation across individuals in such an extreme way. So I think going forward this is going to be a really interesting area to try and think about how to integrate the data sets that we've generated in sepsis with some of the data sets that are currently coming out on the COVID.
Thank you.
Thanks. I know earlier in your talk you talked about pediatric sepsis, and we've got a question about whether the pediatric sepsis also has these similar SRS subtypes, and is there an immunosuppressed subtype as well?
Yeah, so historically it's been quite challenging to try and replicate these pediatric subgroups in the adult patients, but the authors of the pediatric study have looked within our adult cohort, and they do see some similarities between their pediatric subgroup A and our SRS1, I believe is the way around, and pediatric subgroup B and our SRS2. I think what's interesting, we've obviously got completely different gene sets that we're using to define our two subgroups, and they saw some modest correlation if they used their gene set on our data. There's definitely a sort of a big sort of part of the work that we need to do over the coming years of really sort of understanding the sort of consensus clustering, as it were, of how to identify the same group of patients across all these different cohorts and across pediatrics and across adults and things like that. And there are other groups that we're collaborating with who have adult cohorts. So for example, the Mars Consortium based in the Netherlands, they've identified 4 clusters from their adult patients, and we see that one of them is quite similar to our SRS1, and there's a second one that's quite similar to our SRS2. So I think consistently we're all picking up some features that do involve immunosuppression and poorer outcome, and now as a field we're going to have to sort of think about how do we best identify those subsets of individuals. The other sort of interesting angle on this is that obviously we're talking about clusters here and assigning somebody into group A or group B or SRS1 or SRS2, but actually that may be somewhat arbitrary in terms of the cutoff that somebody should be in one group or another. And the other interesting thing to think about is whether or not we should be treating this as a quantitative trait. And so someone should be on a scale sort of with, immunosuppression at one end and immunocompetence at the other. And we should then be sort of figuring out where on that scale that person is, and whether that can then be used in a clinical setting, sort of in conjunction with the clinical data that's collected already and other comorbidities to then help sort of figure out the best treatment plan for that individual.
So we have now a more technical question. How are you controlling for environmental effects in gene expression data for eQTL mapping? Is it PCA? If yes, do you think you may regress the SRS signal out?
Yeah, it was definitely a really good question. So for the work that we've previously published, which was a small eQTL study on the 265 individuals, we did use PCA, and so potentially we were regressing some of the SRS signal out. At that point, we were more focused on just generally looking at the eQTL effects. With this new cohort, we're really interested in looking at interactions. So whether or not the eQTL effects are different between the two groups. And so what we're planning to use for this new cohort is peer factors, so then we can hold out the SRS group when we calculate the peer factors on the remaining variation of gene expression. And then we can just include SRS group in the model. So hopefully then we shouldn't be regressing it out with our covariates that we're using to correct for all the other confounders.
In your presentation, you talked about the scale and the number of patients in the UK, but there's been very few successful trials. I know you're starting this large cohort now to try and get more and more answers. But so, what have been the challenges to now that have stopped progress in a way?
So I think on the clinical trial side, one of the big challenges is the heterogeneity. So sepsis is a clinically defined syndrome, and when you look at the definition, it can encompass such a range of individuals. And the SRS groups is sort of one line of evidence that's highlighting the fact that there actually could be quite a lot of variability between individuals. And so clinical trials have been challenging in that often one trial shows a positive outcome, it's repeated, it doesn't show a positive outcome, and it's really hard to know if it's just because we haven't got quite the right group of patients being enrolled into that trial to really identify who are the right people that we should be treating. And so I think that's the area that we're quite excited about. And again, our collaborators who are leading these clinical trials, we're working with them to sort of think about how and when we can make use of these sorts of signatures in that setting. So whether it's sort of retrospectively, like the VANISH study, retrospectively going back once the trial's complete and being able to stratify patients and start to sort of disentangle whether or not there are potentially differences in the way that individuals are responding. And then, the sort of long-term aim eventually to then potentially have it as part of the clinical trial where people are then assigned to different treatments based on their SRS or similar sort of endotyping based on transcriptomic data.
Thank you. So up to now, you have been using blood instead of biopsies from tissues, I guess, because it's easy to scale. Yeah, but we were wondering if do you think like collecting biopsies for a subgroup of patients will help on the interpretation of the functional data, or even dig into further markers that can be better help on the prognostic for those patients.
Yeah, I think it would be amazing if it's possible. It's definitely just another more challenging step in terms of trying to get access to samples. And, you know, obviously there is an argument about whether or not the blood is the most relevant tissue to be looking at in terms of these signatures. I think the flip side of that is not only the fact that the blood is a much more accessible tissue, which means this kind of cohort size is achievable. It's also trying to think sort of further down the line on the translational aspect, that obviously if you can get any transcription or biomarkers that are helpful either for trying to stratify patients up front before treatment or for monitoring, patients are not too worried about giving a small blood sample. However, taking biopsies is going to be challenging, and particularly if we wanted to do this over time, like, you know, you could imagine if the technology is there, that this could be done sort of every day or two in the ICU to sort of make sure people are on a sort of trajectory that's towards a better outcome, and you wouldn't want to be biopsying them regularly. So I think it would be a fantastic thing to sort of supplement what we're doing, but also if we are able to sort of disentangle the biology from the blood, then I think there's a real sort of translational potential there.
Thank you.
Thank you very much. I believe we've exhausted all the questions so far. They keep coming in, but I think we've exhausted them for now. So I just want to say thank you once again, Emma, for your presentation and for joining us for this question and answer session, and also for the audience that have been pinging in all these questions to us for the last half hour. And next, as you know, there's a Sanger seminar each month, so please look out for announcements and continue to check out the website. Next month's speaker will be Professor Mark Blackster, who's head of the Institute's Tree of Life programme. His seminar will cover diversity genomics and the Darwin Tree of Life project, which aims to sequence all of eukaryotic species in the UK. So the date for your diaries is the 20th of October at 4 o'clock. So thank you once again, Emma, for a brilliant session today and for answering the questions in such a illuminating way. We really appreciate it and look forward to seeing you all again next month. Thank you.